Okay, hello folks. I'm just finishing off the lecture on, uh, what is the lecture? On sampling experimental design. Okay, so we've been talking about uh, different ways in designing an experiment. We've gone through the 10 or so steps. We've talked about sample units. We've talked about how to determine the number of sampling units we want to do. We've talked about how we lay those sampling units out in sampling designs, and we saw there were regular systematic ones, random ones, transects if there was a gradient, an obvious gradient, and stratified random designs of our sampling where we can arrange our sample units randomly in in predetermined areas which you are looking at to see whether they're different or not. Okay, so this might be savanna and this might be forest, for example. Okay, so I want to talk a little bit about the different types of sample units now. Okay, we talked a lot about plots and plotless units, and this is going to be the topic in the first practical, in particular discussing how we can use plots and plotless units when we're sampling trees. Okay, so quite often plots are used as a sample unit. What is a plot? Well, a plot is a defined area in which we take our sample. So we take a small piece of the overall population in a smaller area, okay, and we measure whatever species or community that we're looking at within those plots. Traditionally, they were based on a quadrat, which was a square shape, but they can be found in all sorts of shapes, although squares, rectangles, and circular plots are the most common. And in fact, a transect is often thought of, or can be thought of, as a plot, but a very long, thin, rectangular plot, particularly if you're making observations and measurements to a certain distance each side of your transect line. Okay? The size of your plot or quadrat is very important. Okay? That will determine how much of the population you're actually sampling in each of your quadrats. And it's always a good idea to do a sample effort curve or a sample area curve before you decide on your quadrat size, so that you can be sure that you're identifying and collecting all the species at any one sample site. Okay, so this is an exercise we do with students in first year ecology. Well, I think it's second year ecology now, and you've probably done the exercise where we do a sample effort curve on the Arepo savannas or on some other grassland. I think it's being done mainly on campus these days. And we increase the area of the plot which is measured until we don't get any more new species. And that area of the plot then is taken to be the best size of the plot to get all the species at that particular location. Okay, so on the Arepo savannas, that size of plot is usually about 1/2 meter by 1/2 meter, so 50 centimeters by 50 centimeters. Okay, the good thing about plots is it's very easy to get an abundance measure. when you're using a plot, because you've got a known area, and if you're, say, measuring the number of individuals of a particular species within that known area, you've got a very good estimate of density. Okay, so plots are very useful, particularly when you're doing some sort of quantitative or abundance measure. We can also use plotless methods. Plotless methods can also be quantitative, and they can also be used to give you an abundance, but you need some more tricky mathematics to work that out. Okay, some of the plotless units which are used most commonly are points quarter sampling for trees. So instead of a sampling unit being a plot with a defined area, instead with a plotless unit, the sample unit is a point, and then you measure the distance to the nearest trees, and using an algorithm you can work out the density of trees from those distances. Okay? When it comes to animals, mark, release, recapture methods are also used. They again have to be calibrated against more plot-based methods, but once they're calibrated, you can use these mark, release, recapture methods for animals, which are mobile organisms. Okay, so what sort that sort of measure would be, say, if you have a net and you are catching birds or bats, and you put a ring on them to mark them and then let them go again, and if you catch them again, you can record how often you catch them, how long it takes to, how many other individuals you get before you start recapturing. In that way you can work out an estimate of abundance. Okay, so plot sample units and plotless sample units are quite often used. Plot sample units tend to be used more for smaller organisms and less mobile organisms in terms of animals, and you can use plot methods for smaller plants as well, like the shrubs and herbs, but you can also use plot methods for trees as well, but the plot methods in that case have to be fairly big, so something like maybe a hectare in size, 100 meters by 100 meters, and it can take about 2 days, maybe 3 days if you're in a rainforest, processing and measuring all the trees in a 1 hectare plot. The good thing is, though, that you can mark them permanently and go back and visit them once again in the future. Okay? Trees quite often use point, center, quarter methods as well, okay, as a plotless method. Okay. I should point out that there are 2 different types of sampling that you can do, either qualitative or quantitative. Qualitative sampling is much more arbitrary, and you can't get any measure of density. Quite often, if you're only interested in species presence or absence, qualitative sampling is all you really need. So you can just go to a site, a sample point, a sample area, and you can basically say, okay, that species is here and that species is absent. Okay? You won't be able to give you a measure of abundance, though, but at least you know whether species are there or not. For a measure of abundance, you need to count individuals. Okay, and that would be quantitative sampling. All right? You probably wouldn't use quantitative sampling for organisms which are too small or too numerous to count, like huge numbers of millipedes in the example here. Okay? Or it may be large numbers of grass plants, individual grass plants in a savanna. Okay, you may just want to record presence absence in that case because it's just simply too time consuming to count every individual. Okay, so you'd have to leave that to quantitative sampling to count every individual in a sample plot, but that would give you a very good measure of density. All right. So sometimes when would you use qualitative and when would you use quantitative sampling? Qualitative sampling, as I said, is mainly used when you want to just look at species presence or absence. And that can be quite useful, okay? If you don't labor yourself with any sort of counting of individuals, okay, you can have a method which allows you to take very quick observations at any one sample point. You just go to the sample point, look around, and write down all the species that you see in that particular area. Okay? With quantitative sampling, you would probably take 2, 3, 4, up to 10 times more time taking the sample because you're actually counting the number of individuals. All right? The biggest plant survey that I know of in Trinidad was carried out using qualitative sampling. Okay, the sample points were laid out across Trinidad and visited, and at each sample point, the workers just looked around and dug up or found a representative of every species within that circular plot, a roughly circular plot around the sample point. Okay. All right, let's move on. So sampling data, okay, can be semi-quantitative, maybe looking at a number of organisms or a number of individuals per unit time. Okay. Now, you can see problems with that because it's really going to depend on how fast you count that you are going to get a different number of organisms. So somebody who counts very fast will tend to get a larger number of organisms or a higher density of organisms in their sample point, as opposed to somebody who counts much more slowly. Okay? So that means that semi-quantitative data really cannot be compared with other workers using the same method. Okay, you can only really compare observations made by the same counter because we must assume that they are counting at the same rate each time. Okay? So watch out for that. You may actually be drifting into semi-quantitative sampling without actually realizing it. And that could be a bit of a shock when you come to the end and your examiner says, no, this was all semi-quantitative, so you can't compare your densities with anybody else in the world. Sorry, fail. Okay, and that's never a good thing. So watch out for your sampling methodology or your sampling units drifting towards the semi-quantitative. Try and make sure that you're using some sort of calibrated instrument. Okay? Okay. All right. All right, this is just introducing some of the statistical analyses. The most popular ones tend to be regression designs and ANOVA designs. So regression designs are used more to take a look at species and how abiotic factors or characters of a habitat control the density of species. Okay? We can also use non-parametric types of those, or types of analyses which don't require the residuals to be normally distributed. Okay? And those could be things like general linear models, okay, which are similar to ANOVAs but tend to be taking over from ANOVAs these days. Okay, there are many different types of ANOVAs and many different types of general linear models. Okay, and they are appropriate to use in different occasions, and I'm not going to go into all of the different ones here, but when you come to do an experiment, an experimental design, before you finish designing your experiment, you must have decided on what sort of statistical analysis you're going to do, and make sure that your experimental design or your sampling design does not violate the assumptions based on these statistical tests. All right, so ANOVAs require that all of your sample units be independent, and that the distribution of the abiotic data and the biotic data is normal. It has a normal distribution. Okay? There's lots of different statistical methods. We've got a whole course on biostatistics, so you probably are very much aware of that at the moment. But the point here is that statistical tests fit in to experimental design, and you need to consider what statistical tests you're going to be using before you lay out your sample points. Okay? All right, so just some additional practical questions that we can ask ourselves. Okay? Some methods about research planning and the scientific method. How can I come up with a question, you may ask yourself? How can I come up with some sort of research question? All right, the first thing that you would need to do is to get out into these ecosystems and look for some sort of pattern which you see repeated, but you don't know what is causing it. Okay? So just get out there, experience the habitat, and try and identify things in that habitat or with a particular species that you want to know about. Another way you can come up with a question is read, do a literature review, and identify where the cutting edge of this particular field is. Okay? And then you can ask a question which other workers seem to be asking themselves. Okay? All right, maybe you would have ideas and topics, but you don't have a project. Okay, so you may have a question. So what you need to do there is you need to try and come up with some sort of prediction. Okay? If you have a prediction, then you can develop a question. And once you have a question, you can refine that to make it into a testable hypothesis. Okay? So if you see a pattern, but you don't know how to turn it into a project, okay, think about how you can reduce or take part of that problem maybe, or you can read up on the problem and see how other people have approached it. Okay? So you've got a a question and you want to refine it so that you can find an answer. And that's a very good point, because quite often a phenomenon that you see, like all the birds congregating in one tree, it's a phenomenon, but you don't know what question that you're asking. Okay? How can you turn that phenomenon, that observation, into a question? Okay? So you may ask yourself, okay, all the birds are in that tree. Why are they in that tree? Okay? So your question may be, for instance, are birds attracted to that species of tree at this particular time of year? Okay? So there you have a prediction that the birds are going to be found in that species of tree at that time of year. You have a prediction which you can then go and try and test. And how would you test it? You would go and stake out or look at a number of individuals of that particular species of tree around a particular time of year and see whether the birds congregate there. Okay? And to do it properly, you should also observe the species of tree outside of that time of year to see whether the birds are not there. Okay? All right, so maybe you have a problem, but you want to find an innovative way to look at the problem or to solve the problem. Well, that's very interesting. One way that you can find an innovative way is to make sure that you don't copy anybody. And to do that, you would need to do a literature review, but a methodology literature review to try and find out how everybody approached the sort of question that you're looking at. Okay? And once you found out how everybody looked at it, then you can say, all right, well, let's try it this way instead. Maybe look at new technologies and so on. Okay? I'm always very interested in drone technology because there's so many different questions that looks like we can answer with a drone technology just using those aerial photos and so on. Okay? You may ask yourself, am I making a significant contribution to a body of knowledge? Well, to understand whether you're doing that or not, you again need to read the literature. So is your work, as fantastic as it is, a complete copy of somebody else's experiment which was done 2 years ago? If that's the case, it's not really making a significant contribution. Okay? You need to have something which hasn't been done before. And you can only determine that if you look and see whether in the literature it has been done before or not. Okay? How will I know whether my project is relevant or significant to a theory or a practical question or just a stupid project? Well, again, you have to do a literature search and see whether your project has been done before, whether it's seen to be relevant to a particular research question or not. Okay? It's always good to ask other people. Okay? That's why universities give project students a supervisor, because a supervisor will be able to tell you whether your project is stupid or not, as the saying goes. Okay? How can I conduct a literature review? How will I know what research is going on? What has been done? What language? And what about research in a foreign language? Okay, how can I conduct a literature review? Well, that's an easy question to answer. You have been writing essays pretty much all throughout your UE career, I'm sure. A literature review essentially is an essay. So you've got a lot of those skills already. You need to be able to access databases and the library has some good databases like EBSCO and even their library database. And that will give you, if you use the right keywords, that will give you a very good overview about what work has been done in the area that you may be interested in developing a project in. How can I know whether my project is too big or too small? All right, a project is usually too big if the sample size that you need will take you far too long, okay, to get finished, okay? Or, the sample design with the sample units is just too big for you to get finished in a period of time, maybe 2 years, okay? So your project is going to be too big if you find out, once you've done a pilot survey, that, hey, it's actually going to take 2 weeks to collect enough samples for my pilot survey, and when I calculate the variation, I find that I need to collect 100 sample units, okay, and it takes 2 weeks to do 10 sample units. So I would have to do 10 weeks of sampling. Now, is that too big or is that not, or too small? Okay, you would need to do your pilot. You would need to say how long it takes to do a sample unit. Then you would need to check the variability and see how many sample units you need, okay, to best represent the population. Once you do the sums from that, and there are algorithms, as I said, little apps which will do that for you, then you can work out how long it's going to take, and you'll know whether your project is too big or too small. Okay? If you find out it's going to take too long, then you may need to reduce the scope of your study. Maybe don't do as big an area as you were intending. If you find out that your project is too small, in other words, you can knock over your sampling in about a week and you've got 10 weeks to play with, then maybe you should look at increasing your sampling design, okay? It's not going to hurt, and maybe you can incorporate different things into your sample design and answer different questions. How can I confine the question so it is a master's topic and not a lifetime of study? Again, the secret is there in how long it's going to take for you to do the sampling. Okay? You may actually, if you are interested in changes over time, you may, however, be constrained in that you need to take years to do this particular study. So for instance, if you want to look at growth rates of trees, usually you would measure the trees once and then 3 years later measure them again, and then the next 3 years measure them again. So you would have at least 6 years there, and that means you can't really do it as a PhD unless somebody has gathered the data already and it is there ready for you to analyze. Okay? How do I limit a project so it can be finished in 2 years? Well, I would recommend that you go through that process of determining how long it's going to take to fulfill your sample design and with all the sample points that you have in it. Okay? And I would recommend too that you make sure that you don't spend 2 years sampling, that you give enough time to read up and make sure you can do your literature review and properly design your sampling design and your sampling unit, okay, so that it is realistic and you have and you have time to do a pilot survey. And then once you've collected your data, give yourself enough time to analyze that data, okay? and then enough time to report on what you find. Okay, so all of those components have to be fitted in to 2 years. How will I analyze my data and write my thesis? That's not a one-size-fits-all thing. Okay, you will need to do your literature searches to see how people who did a similar study analyzed their data, how they wrote up their thesis. You are actually constrained about your thesis writing by particular formats which are acceptable at the UE. Okay, how will I know what to conclude? Well, hopefully the research question that you're asking, okay, is based on the predictions that you make. So if your observations coincide with your prediction, then you can talk about that and say that, okay, I believe that this is happening because of this. Okay, if something else goes, you see something else, then you'll need to step back and think about it and try and decide how you can interpret what you see. Okay? You make a hypothesis and then you try and prove it. What if it is incorrect? Well, that's fine. If your prediction is not correct, then come up with a different hypothesis. Okay? And try falsifying that one again. All right? If we start by trying to prove a hypothesis, but we end up trying to fit a new hypothesis to the data, that's fine. That's science. That's showing that you are exploring reasons for the phenomenon. Okay? Will I collect bad data or data insufficient to answer the question? Well, will you collect bad data? It's really up to you to do your quality control checks as you collect your data. Okay? If you see that one of your sample points just does not coincide with the other sample points, it's quite likely that you've collected some bad data. Okay? If you don't have data sufficient to answer the question, you should be able to avoid that by doing the due diligence and the pilot survey to make sure that you have a big enough sample design and a large enough sample unit. Okay? All right, so I'm going to leave it there. Okay, take a look at some of the rest of these questions. You can find the answer in the reference there, Ford 2000. Okay, and see whether you can answer them after reading the reference. Okay then, I will talk to you later. Bye now.